Hello everyone. Today we are going to discuss about the DNA fingerprinting. It is a lab-based technique where we are able to go for the identification of an individual by examining their DNA. This DNA fingerprinting can be called as DNA profiling or genotyping, or in a very simple sense we can say it as a identity testing. We can say it as a identity testing. Alec Jeffrey was the scientist and along with his co-workers noticed that there are certain sequences in the genome which are highly variable. So he was the one who called these variable regions present in the DNA as a mini satellite. He observed that these mini satellites do not contribute to the function of the genes. Jeffrey recognized that each individual has a unique pattern of these mini satellites. So, as the name is ambiguous, identifying the technique that takes the advantage of difference in DNA sequences. So, on the basis of difference in DNA sequences, an individual can be recognized. An individual can be recognized. So, if we have to go for a DNA fingerprinting, we have to go for first the sample from where we will be able to go for the collection of DNA. So, starting with the things we had to go for first DNA isolation, then we have to use molecular markers, then we with the DNA markers, then we have to go for PCR, polymerase chain reaction, or thermocycler is also called as. Then the product which is formed in the PCR has to go for the gel electrophoresis. Then at the last, it's southern blotting. So starting with our DNA isolation, we have to go for the DNA isolation from blood, tissues, semen, vaginal fluids, hair roots, skin, skeleton remains, or elsewhere where we will be able to go for DNA extraction. Simply for DNA extraction, it may be that we can go for DNA extraction, we can go by kit based method or we can go by manual method by using the protocol given by the William et al 1990. So we have to take sample then by giving different chemical treatments to this sample. Like first we have to take the sample, then on the other side we have to make lysis buffer which should be fresh, then adding the proteinase K which will be able to degrade out the proteins which are present on the which is present in the cell membrane. Then we have to go different centrifugation processes. Then at the last we have to precipitate this DNA by adding cold chilled ethanol. Then after precipitating DNA with the cold ethanol, then we have to air dry this pellet. Then we have to dissolve this pellet in a tris EDTA buffer. When this DNA pellet is completely dissolved in this TE buffer, then we can store this at the sample can be stored at 4 degrees centigrade if we have to use in weeks days time, or if we have to use in future, then we have to store this DNA sample at minus 20 degrees centigrade. For its purification, we have to go for spectrophotometer analysis, which is also called as the quantification of DNA. So the quantification of DNA can be taking the ODs at 260 by 280. If we are taking absorbance, if for if we have to take absorbance for double-stranded DNA at 260, then that time we have to take the quantity of DNA should be 50 microgram per liter. Or when we have to go for the absorbance for single-stranded DNA at 260, that time we have to take 33 micrograms per liter of DNA, microliter of DNA. For single strand RNA, we can take 40 microgram per ml DNA. Then we will take the samples at 260 degrees and 260 nanometer. At 260 nanometer absorbance we can take. So this was how the DNA was isolated and quantified. Now, before, when we have to isolate the DNA, then we have to go for the we have to then take the molecular markers for DNA fingerprinting. The mostly markers which we are using is the restriction fragment length polymorphism, RFLP. So what are these markers? DNA markers or molecular markers? These are the simple sequences of DNA which have been traced to simplify the location on the chromosome and associate with the particular traits. These molecular markers are mostly classified into two main categories: type 1 markers and the type 2 markers. Type 1 markers are those markers for which we are using, we should be able, we are for when the function of a particular gene is known, that type of there we can use type 1 markers. When we have to go for unidentified thing, when we are not having knowledge of the organism, that time there we can use type 1 markers. When we are having knowledge of particular gene, that time we can use type 1 markers, and the example is restriction fragment length polymorphism, RFLP. When the organism is unknown to us, we are not recognizing it, that time we can use type 2 markers, and one of the example is the RAPD, random amplified polymorphic DNA. So rapid, it's also pronounced as the rapid. This procedure was first developed by William 1990 using PCR to amplify or randomly amplify enormous segment of nuclear DNA with an identical pair of primers. So what we do, we take DNA sample, then we use primers of length of 8 to 10 base pair in length. Then RAPD and PCR, this technique is very simple. This technique can be used for the identification of genetic markers at different taxonomic levels without having prior knowledge of the genome. Like if we will take example, if we have to visualize or understand the diversity of the plants, there we can use RAPD markers. What we do, we simply take the primer set, then we amplify. If we will take the example of 5 plants and we are having 5 molecular markers, then as per these rapid primers or rapid molecular markers, they will show different bands, due to which we will be able to go for the identification of diversity in different plants. We will see this in the diagram. We will discuss this again. Now the molecular marker is the restriction fragment length polymorphism. So the RFLP markers were first developed by the Boston in 1918, and this RFLP markers were regarded as the first shot in the genome revolution, marking the start of an entirely different era in a biological science. This was the first marker which were developed. So DNA fingerprinting pattern produces by restriction enzymes are high molecular weight DNA fragments which vary from one species to another. So, in this restriction fragment length polymorphism, we are using different restriction enzymes like BAM, H1, ECO, or 1 Henry. Like that, we are having many enzymes. These enzymes are isolated from the different microorganisms. So, when we are having what we do in the RFLP, we simply take the genomic product, means we simply take the DNA, then we incubate this DNA with the restriction enzymes, then allow it to, then when there is a restriction digestion of the genome by the help of the restriction enzyme, then we will allow it to run on the agarose gel electrophoresis. Even for RFLP we can use PCR product as well, but the PCR product is again incubated with the restriction enzymes, then that PCR along with the restriction digested product will be then run on the agarose gel electrophoresis. Then from this age we can go shift to the southern blotting. This is one of the another techniques which can be used for the restriction fragment length polymorphism. Via the southern blotting technique, we will be able to know the copy number of the genome. Now we have to discuss about the another component of PCR, polymerase chain reaction. To start with the PCR polymerase chain reaction, if there is a small amount of DNA available, we are able to amplify it 1000 times, 10,000 times with the help of the machine. That machine is called as the PCR polymerase chain reaction. This is also called as a thermocycler. So for this technique, what we had to do, we had to take a DNA sample. We have to, so that this DNA sample has to be amplified or replicate under in vitro conditions. For this in vitro replication, we need primers. With the help of these primer, nucleotide will get attached, DNTPs, along with the Taq polymerase. This Taq polymerase is one of the polymerases which has been extracted from the Thermus aquaticus. This enzyme is having ability to withstand with the high temperature. When we are having the amplified product, now we can go for either RAPD or we can go for the RFLP technique. So what we do, by the PCR, with the help of by the PCR, we will be able to get 1000 copies of a particular genome. So there are the main 3 steps which are involved in the PCR reaction. First is denaturation. When DNA is getting denatured, the 2 strands are separated up at a very high temperature. Mostly this process is carried out at 95 degrees centigrade. Then is the annealing of the primers at each strand, as you can see here. Annealing of the primers. This annealing of the primers takes place at 55 degrees centigrade. Then the extension, then the addition of the nucleotide to this primer. This reaction takes place at the 72 degrees centigrade. After 33 cycles, we are having bulk quantity of DNA in our hand, which can be visualized by either RAPD or either by the RFLP technique. So in DNA fingerprinting particularly, we use restriction fragment length polymorphism, where we are getting different size of fragments, as we can see here. What we do, assume that we are having mutation in our gene, Due to this mutation, we have lost our restriction site. Due to this mutation, this normal gene has converted into the diseased gene. As you can see in the diagram, the normal gene, this is having 3 restriction sites where restriction enzyme will be able to cut this DNA. So in the diagram, we are having MST1 restriction site. This MST1 has 3 restriction sites on the genome, which by having 3 restriction sites we will be able to get 2 bands. अगर हम लोग एक लकड़ी लेंगे, उसको 2 हिस्सों में, मतलब साइड, एक तो साइड में कट है, एक तो साइड में और एक बीच में, बीच वाले जो रेस्ट्रिक्शन साइट है वो क्या करेगी, इस फ्रेगमेंट को 2 हिस्सों में ब्रेक करेगी. when this normal product will be allowed to run on the agar gel electrophoresis, this product will be able to show 2 bands here, right? Or assume another case, this is about the specific gene. When we have amplified this gene, then we can go for, as I said earlier, we can restrict our either genomic DNA or a particular PCR product. For example, here I am talking about the PCR product. This PCR product is having 3 sites for the restriction site for MST1. So 2 sites are at the present of the ends of the gene and 1 is present on the center. When we go for allow it to run on the gel electrophoresis, there we will get 2 bands. If the middle site is mutated, which is responsible to cause the disease, then the center site for restricting enzyme is lost. Whereas when this PCR product is to run on the gel electrophoresis, there we will get only 1 band because the center restriction site is lost. This was a simple for particular gene product. Or if we are having genomic product, so there will be many restriction sites. So there will be, as you can see here, we are having different bands. Assume we are having 10 to 15 bands, and for disease bands we are also having the same bands, but we will be using southern blotting technique. So what we do in the southern blotting technique, we simply allow our product to run on the gel electrophoresis like here. Then after gel electrophoresis, what we have to transfer to the membrane filter by southern blotting technique. We will be able to transfer our particular gene onto the nylon membrane. So what we do here, we take a southern blotting apparatus where we will be able to get hybridized our particular gene of interest onto the nylon membrane. Then by the autoradiography, we will be able to visualize how many copy number of a particular gene is present in that genome, or we will be able to identify the modification of any particular gene. Like here we are having MST1 restriction site, which has been modified by mutation, due to which this particular gene has lost its ability to get restricted or to get broken with the help of restriction enzyme on the nylon membrane. We will be having only one copy of, or we will be having single band for the particular gene. So what we do in the southern blotting technique, we simply take the gel, then onto the gel we take 2 to 3 simple membrane filter, that will be nylon membrane filter. Then on to the nylon membrane filter we will be able to keep the paper towels. Then above the paper towels we will keep some weight, so by which there will be the DNA fragments transferred exactly as they were arranged in the agar gel. As you can see in the figure, they are exactly opposite to the what they are present in the electrophoresis apparatus or age. The DNA fragments complementary to the probe are visible after autoradiography or chemoluminescence. By this way, we will be able to confirm the gene number or the mutations which are present in the genome. This was how the restriction fragment analysis was carried out. We are having most important application of the RFLP or the DNA fingerprinting in case of forensic science, or either this DNA fingerprinting can be used for the paternity test. Like in case of here, we are having example of paternity test. We are having 5 adults and we are having 3 children here. So the banding, by matching the banding pattern of the adults and the children, we will be able to confirm that who is the father of child 1 among all the adult 3 and who is the father of child 2 and child 3. If we will take example of adult 1, the adult 1 is having 4 bands and the child 1 is having 4 bands. So if we will see the location exactly, the adult 1 is having first band which matches with the child 1 and child 3. Then again the child 1 and child 3. Then again the second band of adult 1 is present in, not in the child 1, not in the child 2, but it is present in the child 3. So 2 bands of the child 3 are matching with the adult 1. Then again we can see the things. Then the child 3 is not having any band resemblance with the adult 1 and the child 3 is having resemblance of 2 genes with the adult 3 and the child 1 is having 1 band similar to the adult 1. So if we will, this is a little if we will take another example where we will be having, by this technique we will be able to confirm which child is who. But the maximum number of bands which is getting matched to any particular father is the father of that child. So this is the how paternity test is conducted. Like in simple case we can say that the एक बच्चे के 5 बैंड्स है और अज्यूम करते हैं कि मैक्सिमम 4 या 5 बैंड किसी पर्टिकुलर पेरेंट या फादर से मिलते हैं, then that child belongs to that father, and the 1 band which is not matching to this father have been come from its mother. So here what we have seen, that child 3 having similar, how many bands of similarity? 2 bands of similarity with the adult 1. Now we will take another example, the child 1. The child 1 is having similarity band of adult 1 for first band. For its second band, this is matching with the this is the for child 2, that child 2 is having matching of 2 bands with the adult 3. Then the child 2 again is having similarity of band with the adult one and the rest is not present there. So by this way we will confirm that which parent or who is the father and who is the mother by comparing the maximum number of matching of the bands between the child and the adult. So the another marker which is there is the RAPD, random amplified polymorphic DNA. So band 1, or we can say val 1, val 5 and val 18. There we have used 7 different primers. In all the 3 cases we are having same primers used. Now we will observe that the restriction pattern